So now I'd like to introduce Dr. Marina Picciotto. Dr. Picciotto is the Charles B. Murphy Professor of Psychiatry and Professor of Neurobiology and Pharmacology at Yale University. She received her BS from Stanford University and her PhD from Rockefeller University under the supervision of Dr. Paul Greengard. She went on to conduct postdoctoral work at the Pasteur Institute in Paris, and she joined the Yale faculty in 1995. So the goal of Dr. Picciotto's lab is to understand the role of single molecules and complex behaviors related to psychiatric illness. More specifically, she has focused on understanding nicotinic acetylcholine receptors, which I believe she'll be talking to you about today. She's used multiple methods and multiple approaches to studying these receptors, including knockout and transgenic mice, viral vector-mediated gene transfer, pharmacology, and behavior analysis. More recently, some of her work has focused on the interactions between stress and nicotine and the role that those nicotinic receptors play. She's been able to model many of the behavioral effects of nicotine in mice and demonstrate the links between individual nicotinic receptors and stress-related effects of nicotine. In addition, she's also focused some of her research on the neuropeptide galanin in opiate reward and withdrawal. And overall, these studies have helped explain the molecular basis for complex interactions that lead to addiction, stress interactions, and may help target new medications. Dr. Picciotto, I'm behind a slide. Dr. Picciotto is also, or was also, the senior editor for the Journal of Neuroscience from 2006 to 2012, and she sat on many NIH and other study sections. She's been the recipient of numerous awards, including several NARSADs, and most recently was an elected fellow at the Institute of Medicine. This is a very important position, as the Institute of Medicine asks and answers the nation's most pressing questions about health and health care. The Institute of Medicine, if you don't know, is an independent, nonprofit organization that works outside of the government. Works outside of the government to provide unbiased and authoritative advice to decision makers and the public. Members are elected for their excellence and professional achievement. For those at the top of their field, membership at the Institute of Medicine reflects the height of professional achievement and commitment to service. Additionally, she has fostered and mentored many students throughout their achievements, such as National Institute of Mental Health Research Awards and training grants. She has over 174 publications and 6 more currently in press. As Quinnipiac grows its own commitment to neuroscience and seeks to attract new students, I particularly admire Dr. Picciotto's unique ability to make science accessible to wider audiences. This is evidenced by her role in BioStep, which is an intensive summer research program for students in underrepresented groups who are interested in biomedical research. We're extremely pleased to have Dr. Picciotto speak to us today, and so please join me in welcoming her for her talk.
So first of all, thank you for that very generous introduction. I am actually relieved now that I've seen pictures of myself that I chose to wear a different jacket than the ones that are in those pictures. That would have been highly embarrassing. So before we start, I'd like to thank the organizers for putting on this incredible meeting. So let's thank them, because this was an incredible amount of work. Great job. And second, I would like to welcome you, and thank you for coming out on a Sunday morning, which is pretty awesome. It's not every profession can get a whole bunch of people out on a Sunday to come listen to neuroscience. So go neuroscience! Another, I think, round of applause. So I'm going to talk to you today about an overview of basically our philosophy of how we can get from very basic science to really important questions for human beings. And overall, although I'm going to be presenting the research that we've done, what I really want you to pay attention to is the fact that we can use molecular approaches, we can use techniques that are really asking fundamental questions about how molecules and neurons work, and then we can actually broaden our approach to take in behaviors that are important and ultimately to investigate in human subjects how we can use this information to develop new treatments or to go forward in understanding human disease. So I'm going to give you one problem, but in fact, of course, this is applicable to many, many of the projects that you're probably working on now, and I'm happy to talk more about this approach later after the talk. So here's the problem that I'm going to talk about today, and that is that there are still, despite the rise of obesity, the number one preventable cause of death today in the developed world is still smoking. We still volunteer ourselves to do a neuroscience experiment on the effects of nicotine on our brains, and up to 350,000 people die in the United States alone every year due to smoking-related illness. And of course, this is a medical problem. Lung cancer is something that we can investigate. We can investigate chronic obstructive pulmonary disease. But wouldn't it be easier to understand the neuroscience of the behavior that drives that addiction and that keeps people smoking, despite the fact that every one of us in this room knows that it's a really, really bad idea? So the majority of smokers actually would like to quit. And I was at a conference years ago when I was listening to a panel of teenagers who all said, yeah, I started to smoke somewhere between the ages of 13 and 16, and then I got to 18 and I really thought this was stupid and I decided to stop, and I couldn't. I made that decision before I could actually figure out that later on it would be a regret. And when it came time to stop, I actually could not change my behavior. There's the idea that this is a failure of will, that it's a problem with a habit, and that all you have to do is really make the decision to stop. But of those who really choose to stop, if they do it without any help, if they just say, I'm going to go cold turkey, only about 6 to 8% actually can still remain abstinent from smoking a year later. So this is still a current problem. It used to be that probably around 60% of the population smoked. Now it's down to about 25 or so, maybe somewhere around 23% of the US population. So the people who were able to say, okay, well, I just do this for fun, have probably stopped, and those who are still smoking are very dependent on nicotine. So let me outline now the molecular, biological, neurobiological problem that we're working with. So nicotinic receptors in the brain, and I'll talk a lot more about this, are distributed very widely. This is an image through a human brain showing you that here's a structural MRI showing you the brain areas, and here's an overlay of binding of a nicotine-like molecule to various brain regions. And you can see actually that here there's a very high level of binding, there's a little bit lower here. These darker blue areas are still actually significant areas where nicotine binds in the brain with high affinity. And in fact, for human imaging of these receptors, it's quite remarkable. It's very hard to get a negative control. There isn't a brain area you can go to and say, well, let's subtract that, because these nicotine receptors are in almost every neuron. And then there's this other problem. There's an incredibly wide variety of reasons that people say that they smoke. And I'll again go into some of this in greater detail. But if you ask one smoker, they may say, well, I just enjoy it. And what does that mean? That means that the reinforcement, the parts of the brain that are in charge of drug reward are being activated by nicotine. There are others that say, well, I smoke to manage my anxiety. I smoke to manage my depression. It's another set of brain areas and another set of behaviors. And finally, there are those that say, well, I'm afraid to gain weight when I quit. And I will talk a little bit more about all three of these. And in general, what we want to know is, are people smoking for the same, to activate or inactivate the same nicotine receptors? Are they smoking to activate or inactivate the same brain areas? And are they smoking to activate or inactivate the same behaviors? And the answer to all of those is, nope, it's not the same. There are a lot of reasons. And finally, here is the big problem that we are actually interested in approaching, and that is that there are an awful lot of nicotine receptors. So this is a family tree of all the nicotine receptors. You can see that there are about 15 different subunits that have been identified in human and mammals in general. This family up here are those that are at the neuromuscular junction. That is, that all of your significant communication between your nerves and your muscles actually go through nicotinic acetylcholine receptors. And they use the normal neurotransmitter acetylcholine. Nicotine does not act very well at these muscle receptors, which is good, because then, in fact, we would really have trouble with movement. But it's this family here, these receptor subunits here, that are expressed in neurons and throughout the brain that we think are absolutely critical for the behavioral effects of nicotine. So here's a little bit more about these receptors. Those of you who are molecular biologists will probably recognize these because they're sort of the poster child for excitatory, amino acid neurotransmitter-gated, ligand-gated ion channels. So here's what they look like. They span the membrane. They're large proteins, and they have a pore right down the middle. And when acetylcholine, the normal neurotransmitter, binds to these receptors, the pore opens, ions flow into the neuron, and the cell is depolarized. Nicotine binds at exactly the same site as acetylcholine, well, exactly as in the same part of the molecule, not necessarily exactly the same amino acids, and it does the same thing. It opens these receptors. But what is really important to remember about these receptors is that whereas nicotine can open these receptors transiently, it can then desensitize or turn off these receptors for a much longer time. So although the initial thing that nicotine does is to activate neurons to open these channels and depolarize neurons, over a longer period of time what it does is to interfere with the normal signals through acetylcholine. So when acetylcholine is released, if nicotine's been around and desensitized these receptors, it can no longer do what it normally should do. These are sort of head-on diagrams of what the nicotinic receptors look like. They're like staves around a barrel. 5 subunits are arranged around that central pore. And what's important to remember is that acetylcholine and nicotine binds at the interface between these subunits. And what's important about that is when you have a large family of subunits that combine in different organizations, each of those interfaces is going to have slightly different properties. Each one is going to bind a little more tightly or a little less tightly to nicotine or to acetylcholine. Each one of those is going to make the channel open for longer or a shorter period of time and maybe make the channel more or less likely to desensitize after it's open. So it matters which of these different subunits are arranged in that receptor, and it's also important for drug development. If each of these interfaces is different, then you can imagine that we could actually make medications that would target different interfaces differentially. And I'll talk a little bit more about that at the end. So here are the family of receptors that are important in neurons, and you can see that there are 3 broad types. I've diagrammed just 3 types, but you can imagine that with all of those subunits, this is really just a broad representation, and the 3 families are those that are homomeric, that is 5 different subunits are exactly the same around the outside. So they are going to have very different properties from these, where you need at least 1 alpha and 1 beta to make a functional receptor. And if the nicotine binds at the interface, you can see that in these types of heteromeric receptors, there are going to be 2 binding sites, whereas in these homomeric receptors, there are going to be 5 binding sites, and that's going to change how these channels react to nicotine. In the brain, we have mainly these 2 families, but this family is represented in a smaller subset of neurons. But in the fight or flight response, in the autonomic ganglia responsible for example, the adrenaline response to stress, to fear, this is the receptor that's really critical for the fast synaptic transmission, for the ability of acetylcholine to drive those ganglionic responses. In the brain, these are more modulatory, so they change behaviors that are already initiated to some extent, rather than driving behavior the way that these autonomic ganglia receptors do. So in terms of where they're expressed, here's where I showed you before, where nicotine binds in the human brain, and in rodent and in other mammalian brain systems, it's pretty similar. You have a very high level of receptors in, for example, here's the thalamus, which is the part of the brain that's important for relaying sensory information to the cortex. And then there's 2 families of receptors. I don't know how well you can see this, but you can get an idea that there's some subunits that are really broadly expressed, and they follow this pattern of nicotine binding. And there's some that are really localized, like these here, that are only expressed in this case, these are the dopamine cell bodies in the midbrain that are important for the rewarding properties of drugs of abuse. And that just simply sets the stage to tell you that we can use perhaps pharmacological agents that target these different subtypes to get very specific effects on behavior or more general effects on behavior. And we're going to now segue into the many reasons that people smoke. So the first really important reason is that people get addicted. And despite the fact that actually tobacco industry executives still say, well, you know, people choose to smoke, it's not an addiction. There's evidence from human studies, there's evidence from animal studies that in general, nicotine activates the same parts of the brain as other drugs that are abused by humans, including cocaine, amphetamine, morphine, heroin, and that drives the rewarding aspects of cigarette smoking, and that those kinds of rewarding aspects allow you to make associations between that reward and your environment. That drives compulsive behavior, habitual behavior, which is not habit, but in fact the ability of cues in your environment to take over and drive your smoking intake. So I showed you this, and now you can see it a little bit larger. But here in the midbrain are the neurons that are activated in some way or another by every one of those drugs of abuse. Any addictive substance that is used by humans somehow increases dopamine signaling from this area. So here in the middle, this is a mustache, right? It looks really very characteristic. If you see it, you know you're in the dopamine system. And what you can see is that here in this medial region is the ventral tegmental area, which is critical. If we ablate that area or if we block it, then drugs of abuse no longer actually can control behavior. And what you can see is that there are a lot of nicotine receptors in that brain area. If you look here are those very selective subunit expression patterns. Alpha 6 and beta 3, doesn't matter what their names are almost exclusively expressed in these dopamine neurons. And then if you look at these broadly expressed subunits, beta 2 and alpha 4, well, they're actually enhanced, enriched in this brain area as well. And there are accessory subunits that are maybe more broadly expressed, but also seem to pull out this area. And what that suggests is that these nicotinic receptors really can control activity in this area essential for drug reward in a way that is very effective. So here is my diagram of a very, very, very highly simplified brain. It has actually 5 neurons in it. And all of those neurons can be controlled by nicotine receptors. And what you can see here is those are the dopamine cell bodies, and I told you they're sitting in the ventral tegmental area. They have nicotine receptors on them, and when nicotine binds to those receptors, it increases the firing of these dopamine neurons. Very simple, increases the release of dopamine, and that can result in the rewarding effects of nicotine. But that's not enough, because there are also other ways that nicotine modulates this circuit. So for example, the excitatory inputs to these dopamine neurons have nicotine receptors on their terminals as well, so that nicotine drives not only the direct response of the dopamine neurons, but actually gives it an extra punch by increasing glutamate, the excitatory neurotransmitter release onto these neurons. And on the terminals, there are nicotine receptors that are enough to induce dopamine release even without action potentials coming down the axon. So it can hit it here, it can hit it here, it can excite it here. What's interesting is that when you're naive to nicotine, nicotine can also increase inhibition onto these dopamine neurons. So at baseline, you have this mixed excitation and inhibition, so you have one set of responses, and then over time, in a slice, nicotine can desensitize or inactivate these receptors while these receptors stay active. So you can go from a mixed excitation and inhibition to a more unmixed drive of this circuit, and that's thought to be one of the reasons that, despite the fact these receptors can turn off or desensitize, prolonged exposure to nicotine, like the kind of prolonged exposure you would expect over years of smoking, can actually go from a mixed excitation inhibition to a more unmixed drive of this circuit. So in fact, over time, the nicotine signal gets stronger in this circuit. So what does that do in a whole organism? Well, if we take slices through those dopamine cell bodies and we look at what nicotine does to the firing rate, normally it increases the firing rate of those dopamine neurons. And we can start to identify which molecules are essential. Because if we take away one subunit, the beta-2 subunit, which is the one that's responsible for this broad pattern of general binding to nicotine throughout the brain, what you see is that broad binding goes absolutely away in knockout mice that lack that subunit, and so does the ability of nicotine to cause the firing of those neurons. And if we just look more broadly at the ability of nicotine to stimulate the dopamine system by evaluating the release of dopamine in the terminals of those ventral tegmental areas, what you can see is in a normal animal, we have a dose-dependent increase in dopamine release when we give nicotine to these peripherally, and that completely goes away if we take away one subunit out of those 15. And that goes along with the ability of nicotine to be rewarding. We can actually evaluate this by asking, and I call this the bar office experiment. If I gave you permission to go to the bar and smoke and drink as much as you wanted, or if I told you when you go to the office you can't smoke or drink at all, and then on Saturday I asked you to choose, where do you want to go? Do you want to go to the bar or to the office? Those of you who actually do smoke or drink, or who have other rewards, let's say food, would probably go to the area where you could get those rewards, and you would avoid the area where you are forbidden from having those rewards. And that's the same as this place preference experiment. What we do is we pair exposure to nicotine with one compartment of a chamber, and exposure to saline, just a random non-rewarding stimulus, in the other compartment. And after training, we ask the individual to choose between those compartments. And if they liked the nicotine, they'll go spend more time exploring the compartment where they got the nicotine. And that's what you see here. At baseline, both compartments are exactly preferred the same way. And after training with nicotine, there's more exploration of the nicotine-paired compartment, less exploration of the saline-paired compartment. And when we take away one subunit of the nicotine receptor, that completely goes away. Nicotine is no longer preferred, and in fact, saline and nicotine are equally boring. So there's no change in behavior. And we use this as a measure of nicotine reward. And what I can tell you is that we can also start to identify the cell types that are involved, the brain areas involved. As you can see from this picture, if you take away the beta-2 subunit, all of the nicotine binding goes away, but we can put it back using conditional methods to express the nicotine receptors only in particular brain areas. And what you can see here is that in this particular transgenic, we put the nicotine receptor back only into the mustache, into the VTA, and into the terminals. Here's in the forward part of the brain, in the nucleus accumbens, where these neurons actually project and release dopamine. And when we did that, what we could do is to rescue behaviors that are associated with psychostimulants. And so in this case, I'm just showing you one behavior. As a field, this has been done for reward, it's been done for reinforcement, for habitual behavior. In this case, I'm just going to show you psychostimulant effects. So you know, psychostimulants like cocaine and amphetamine can actually arouse a person or an animal, and we can measure that by increased locomotion. And what you can see here is that nicotine does the same thing. When a normal individual gets nicotine, they're actually more active. If we take away these receptors throughout the brain, there's much less, where there's no effect of nicotine on a psychostimulant response. And if we put it back only into the dopamine neurons, we can rescue that. So that means that although these receptors are expressed throughout the brain, you actually only need them here for at least some of the behavioral responses to nicotine. So as a field, this has been a huge success story. Many, many different laboratories have been working on this over the last about 15 years, and using various combinations of molecular techniques and various behavioral tasks, we've been able to narrow down exactly which receptor is involved in these, let's say, reinforcing or psychostimulant properties of nicotine. We've been able to identify the cell types in the brain area, and we've been able to actually target these receptors, not as a drug company has done this, and treat smokers and help smokers who would otherwise be unable to quit. And I'll just give you a quick summary here. We can actually make some of the nicotine subunits hypersensitive, and we can show that that is actually sufficient to drive a preference for nicotine, that reward experiment. I showed you the transgenic expression of one subunit in the VTA, in the ventral tegmental area, and that rescues psychostimulant effects. You can use a virus to re-express a particular subunit in that brain area and show that that rescues the willingness of the animal to work for nicotine. And we can actually knock out a nicotine receptor only in those dopamine neurons and show that that is sufficient to get rid of nicotine reward. And altogether, this has given us the name of the receptor. It's alpha-4, alpha-6, beta-2. It's given us brain area, ventral tegmental area, and shown that that is sufficient for nicotine reward, reinforcement, and psychostimulant effects. So here's our diagram again of our different nicotinic receptors groups. Here's the receptor that has been identified. Here's the brain area that's been identified. And it's this interface right here that's targeted by Chantix, varenicline. I have no association with the drug at all, but Pfizer developed that with an intelligent drug design method to target this interface and find a way to stimulate it a little bit, but block the effects of nicotine at that interface. And that partial agonism is what makes this actually currently the most effective way to help smokers who want to quit get abstinent. So I think that's a success story because in psychiatry we've been trying to find receptors for various psychiatric illnesses. We've been trying to find drugs that very selectively target those receptors and to see if we can actually help people who are otherwise unresponsive or not medicated by the current treatments we have. And here's one for perhaps our most important killer in the psychiatric world, and that is nicotine addiction. So you can imagine that decreasing smoking by about 10% would already be enough to save many, many lives. Okay, so that's one. It's very obvious. It's an addiction story. But why else do people smoke? Well, another thing that people report is that they smoke because they're afraid to stop smoking and gain weight, and even scarier, one group of smokers, teenage girls, report that they start smoking specifically to try to be lean, to lose weight. And if weight control is the number one reason that a particular group of smokers are saying they start, that's really particularly tragic. So is it possible that it's the nicotine in smoking that drives this overall decrease in weight in smokers? You can imagine there's a lot of reasons, right? It could be just that you have something in your mouth, something in your hands. It could be one of the other 4,000 constituents of tobacco smoke that changes metabolism or changes weight. So we have to ask, is it the nicotine in tobacco that's important? Well, there are some clues from animal studies. And for example, the laboratory of Neil Grunberg was a pioneer in this area, and he showed that in rats, at least, nicotine alone can decrease food intake. And there's interesting sex differences. Female rats are more sensitive to this appetite effect than male rats. And Michele Zoli and his group also showed that you can model the fact that nicotine withdrawal can increase food intake, decrease metabolism, and increase weight. He showed not only that the behavioral consequences of nicotine withdrawal matched between rat and human, he also showed that he could find molecules that were associated with changes in metabolism that were induced by nicotine withdrawal. And that all leads to the idea that perhaps it is indeed the nicotine in tobacco that controls appetite in humans. However, in general, the neurobiological effects of these mechanisms are not really well understood. So that's what I'm going to tell you about now. So first, we have to show that we can model these effects in a model, in an animal model, in this case in mice. And what you can see is that mice, in general, gain weight over time, and this is from very late adolescence, about, let's say, 8 weeks of age, all the way through until adulthood. And if we give nicotine to these animals, they actually decrease the rate of weight gain. And you can see that there's a dose dependence so that at the slightly higher doses, there's even a little bit of dip in their weight before they start a whole new curve of weight gain. And these kinds of doses are very, very low. They're not very different from what you would expect if there was heavy smoking. So what can we do? We can now start to use pharmacological agents that selectively target different nicotine receptors and ask, which of these receptors might be implicated. And luckily, there are some pharmacological agents that are out there that we can try. So here's two. Here's nicotine. I've already showed you that that is effective at decreasing the growth curve. And what you can see is that there are all of the heteromeric receptors, in fact, all of the receptors for nicotine, not surprisingly, are responsive to nicotine. And you can see that here in a slice preparation in neurons in the brain, nicotine causes an inward current regardless of which receptor is on that neuron. But here's another nicotinic-like molecule. It's called cytosine. It's an extract of a plant called laburnum, which actually grows outside. It's probably starting to bloom and give you allergies here on campus. And this extract is interesting in that it is used in a smoking cessation aid in Eastern Europe that is called, I don't remember. However, that smoking cessation aid is basically just cytosine, which acts a lot like that Chantix drug that I told you about already. It is a partial agonist of the receptors that are important for nicotine reinforcement. And what does that mean? That means that nicotine causes a big opening of those receptors. Cytosine causes a little opening, but if you combine the two, it blocks the effects of nicotine. At another set of receptors, these are the ones I told you about that are responsible for the fight or flight response, nicotine and cytosine act exactly the same. So does cytosine have the same or a different effect on weight gain? It has the same effect as nicotine. So that gives us a clue. If it acts the same on those alpha-3 beta-4 receptors, first of all, that suggests the receptors that are important for nicotine reward are not the same receptors that are important for appetite. And it also tells us that now we can perhaps target the two receptor subtypes independently. So here we've got another picture of our receptors. This time it's not this heteromeric receptor that's important, but in fact this receptor that has the alpha-3 and the beta-4 subunit that may be important for appetite. So the autonomic ganglia are not in the brain. So if we think that perhaps it is the fight or flight response that's important for this effect on appetite, and you can imagine that it might be, you wouldn't want to go eat if you were being chased by a tiger, then it might be that this effect of nicotine is actually happening somewhere else in the body, not in the brain. And so what we can do is we can block the peripheral receptors, those in the ganglia, those in the body, with a drug that doesn't get into the brain, and we can say, does this block the effect on feeding? And what you can see here is that it does not. So here you see that if we give animals this drug that decreases their appetite, they eat a little bit less. And if we try to block that effect with a nicotine blocker that doesn't get into the brain, it doesn't actually alter that appetite suppression. So in fact, this is probably not happening in the body. It's probably happening in the brain. So where in the brain might that be? Well, we know a lot now about the parts of the brain that control food intake. And what's really cool is that we can now, as a field, drive the firing of different neurons in the hypothalamus and show that there's one population of neurons that actually causes animals to eat and one population of neurons that causes animals to stop eating. And they're both sitting in the same brain area, and this is work by many people, but most recently by Scott Sternson and his colleagues. Here is diagrammed the arcuate nucleus of the hypothalamus. Now, the hypothalamus is a part of the brain that really tells you to match your behavior to the needs of your environment. So what does it do? It tells you when you're thirsty, it tells you when your body needs calories. It tells you, actually, it's also involved in stress responses. It tells you when it's less important to get calories than it is to escape a predator. And all of those signals converge on the hypothalamus, and somehow the hypothalamus balances those signals, and the output says, eat if you stimulate your NPY neurons, stop eating if you stimulate your POMC neurons. So we were very naive. We said, okay, well maybe what nicotine is doing is somehow it's activating these POMC neurons. So how do we test that? Well, one way that you can test it is look at a molecular marker of the history of whether those neurons have fired when you give a challenge with nicotine that actually decreases appetite. And the way that we do that is by looking for immunocytochemistry for an antibody response that recognizes a marker called CFOS. Now, CFOS is simply a gene that is induced by high activity in a particular neuron. So what we can do is we can give animals nicotine, we can evaluate whether or not that decreases their appetite, and then we can actually look for C-phosphamine activity and co-stain for POMC, the marker of the neurons that tell animals to stop, and humans, to stop eating. And what you can see here is images through the arcuate nucleus of the hypothalamus. The dark brown stain is C-phos, and this purple stain is POMC. And when we evaluate and we count the number of neurons, particularly throughout the hypothalamus, that are marked with C-phos after a nicotine challenge, you can see that cytosine and nicotine overall don't cause a change in the C-phosphamine reactivity. But when we co-label only for those POMC neurons, the appetite-suppressing neurons, you can see there is a big induction with both of the nicotine drugs that decrease appetite and the POMC neurons. And what's interesting about that is probably what you're getting if you get no overall change is you're getting a decrease in the activity of the neurons that say eat and an increase in the activity of the neurons that say don't eat. And what you can see that's very important for the validity of this finding is that that doesn't go away over time. Whereas many of the effects of nicotine are desensitized, that is, they decrease over time, this effect stays even if you treat for 28 days. Nicotine still, in this case cytosine, still decreases food intake, and it still increases the activity of the neurons that say stop eating. All right, so this is a potential place that nicotine is acting. This is by no means proof. This is pure correlation, but it gives us a hint of where to look at some more. So the first thing that we're going to do is the same thing that I told you was done for evaluating the effects of nicotine on reward, and that is let's look at a slice through the brain in the area where those POMC neurons sit. And we can do this because we can express a fluorescent marker, GFP, only in POMC neurons. And when we record from POMC neurons, what we see is that nicotine increases their firing. This should look familiar. And that gets blocked by a nicotine blocker. It's dose dependent. Mecamylamine is a nicotine blocker. It completely blocks it. And it suggests that, yes, at least in this slice, we can stimulate those neurons directly with nicotine. So now what we want to do is it enough to eliminate those POMC signals to get rid of the ability of nicotine to control appetite? So how do we do that? Well, we can look at a knockout mouse that doesn't have POMC at all and ask, in those knockout mice, does nicotine have any effect on appetite? And I'm showing you here wild-type mice. Here's a dose-dependent decrease in food intake when we treat with cytosine in this case. And that goes away in these POMC knockout mice. So that's a hint. Okay, good. POMC neurons may be essential for this effect. But because this is just one node in the pathway, and because these neurons are so essential for food intake in general, it's important to interrogate this another way. So what we asked is, can we intervene in another point in this pathway and get the same effect? And so what we know is that these POMC neurons have their cell bodies here. The neurons project upward in the hypothalamus to the paraventricular nucleus, and there they signal through MC4 receptors to send that signal to stop eating, go do something else, you've had enough food, to other places in the brain. So if we can now intervene here by decreasing the activity of these MC4 receptors, is that now sufficient to block the ability of nicotine to decrease feeding? So the way that we did that was to design viral vectors that target a very specific gene using something called a small hairpin RNA, an shRNA, that finds one particular target in the genome, binds to the RNA, and both blocks its translation and causes its degradation. It's a very nice way in cells or in the brain to target a particular gene without disrupting the rest of the genome, and to do it in adulthood and in very specific neurons using a viral vector to deliver these shRNAs. So we delivered these shRNAs to the paraventricular nucleus of the hypothalamus, and we targeted the POMC receptor, MC4. So what we could show is that we could decrease the expression of MC4 very efficiently, and that when we put this shRNA into the paraventricular nucleus, that we could block the ability of nicotine to decrease food intake. So here's a GFP control, so just a control for the viral vector infusion into the paraventricular nucleus. We still get the ability of cytosine to decrease food intake, and that's highly blunted when we knock down MC4. So we have a circuit. We know where we want to look for nicotine's effects on appetite. But we have this strange receptor. It's expressed mostly in the autonomic ganglia, and here we're proposing that it's actually acting in the arcuate nucleus of the hypothalamus to change the activity of POMC neurons and to decrease food intake. So what is the evidence that there is actually any nicotinic receptor of that type in the hypothalamus? It wasn't known. So what we did was to take those POMC GFP expressing sections and to isolate the neurons that express GFP using a laser capture microscopy procedure, to take those cells and to prepare RNA from them, and then to use quantitative PCR on the RNA expressed in those cells to ask the question, is the beta-4 subunit even expressed in those neurons? And what you can see here is that, yes, those neurons have the RNA, and here I'm showing the cDNA, for the POMC gene. That's good. We've got the right identified neurons. They also express the beta-4 subunit, and we know that's selective because if we do this in a beta-4 subunit knockout, that transcript is gone. So indeed, to our surprise, in the arcuate, there is this important subunit that controls the response to stressful events in the environment, and in the brain, this particular subunit is important for the ability of nicotine to decrease appetite. Now, I've shown you it's expressed, but I haven't shown you that it's necessary for this effect of nicotine. So then what we wanted to do was exactly the same. We wanted to use shRNAs targeting only the beta-4 subunit in the arcuate nucleus, and to ask, if we knock down the beta-4 subunit, can we abolish these appetite effects of nicotine? And so as a nice control here, we had another subunit, the one that we know is involved in nicotine reward. It's also expressed in this part of the brain. We could knock that down and say, if our hypothesis is correct, we shouldn't have an effect of knocking down beta-2, but we should have an effect of knocking down beta-4. And that's what we've done. So I'm showing you here again validation that we can actually knock down either beta-2 or beta-4 using these small hairpin RNAs that we deliver with a virus. Here's the arcuate nucleus of the hypothalamus. Our control is a scrambled shRNA delivered with an AAV vector. Here's beta-2 knockdown, here's beta-4 knockdown. In each case, we challenge with cytosine, and you can see that here we have a decrease in food intake in the normal animal. We have a decrease in food intake when we knock down beta-2 in the arcuate, but that completely goes away when we knock down beta-4 in the arcuate nucleus. So really, this is good evidence that the pathway that we've outlined is actually essential in the arcuate nucleus for the appetite-suppressing effects of nicotine. And here's a sort of summary of our circuit, and that is that in the arcuate nucleus we have mingled POMC positive neurons that decrease appetite and NPY positive neurons. There are nicotinic receptors containing the beta-4 subunit on these POMC positive neurons. When nicotine comes into the brain, they're activated. They go up to the paraventricular nucleus where they activate the MC4 receptor on paraventricular nucleus neurons. And this signal is essential for the ability of nicotine to decrease appetite, decrease food seeking, shift behaviors presumably to others that are not involved in this particular behavior, and to cause the weight control aspects of nicotine signaling. So back to this picture, you've seen it before. Now we're targeting these receptors right here. We know they contain the beta-4 subunit. We don't actually know what the alpha is. We know that in the ganglia, in the fight or flight response, it's alpha-3. But in the arcuate, we're now actively trying to ask, what are its partners so that we can target this interface and find pharmacological agents that really work selectively to suppress appetite. And we know the brain area. We know that the arcuate is absolutely essential, as are its projections higher in the brain. So that's another reason that people smoke, but it is, again, only one of many. So I'm going to ask now one more behavior. Why do people smoke despite the fact that they know that it is highly toxic? The reason is, another reason is that people smoke to control their symptoms of mood and depression. So one thing that we know from epidemiology studies is that people who are depressed are much more likely to smoke. I told you about 20 to 25% of the general population smokes. Those who have depression have about double the likelihood of smoking. And we have a paradox, and I'll tell you what that is. In human studies, a number of investigators have delivered nicotine different ways and found that they have very different effects on mood. So Paul Newhouse and his colleagues in Vermont were able to infuse nicotine very rapidly in an IV paradigm, and he found that his patients reported that they felt depressed and anxious. Another set of studies have used nicotine patch to actually treat depression symptoms in people who are never smokers. So in those studies, nicotine delivery is antidepressant. So how can that be? Why would two forms of nicotine delivery have very opposite effects on mood? Well, as molecular biologists, as people who work in model systems, we can actually answer that question. There are hypotheses that we can make. So first of all, we know about the channel properties of these receptors. We know that high levels of nicotine delivered rapidly should activate these receptors, should open them. We also know that low levels of nicotine that are delivered constantly over time are much more likely to desensitize these receptors and actually keep them closed when you give a challenge with nicotine or when you signal with acetylcholine. So based on those human studies, we can make a hypothesis. Our hypothesis is that maybe it's not opening nicotinic receptors that is antidepressant. Maybe it's actually blocking them. And if that's the case, then smokers who transiently open and then desensitize their receptors over the period of the day may in fact be increasing their depression symptoms during the time that those nicotinic receptors are open, and then solving their problem that they've just created by smoking by desensitizing their receptors during the course of the day. And as basic scientists, we have a way of testing this hypothesis because there are non-nicotine ways of blocking these receptors. So I told you about mecamylamine. Mecamylamine is a channel blocker that sits in the pore of nicotinic receptors and blocks their activity. And we can ask, if we treat with mecamylamine, does that actually act like a classical antidepressant? And the answer is, yes, it does. So we can evaluate the antidepressant-like properties of molecules. I'll never tell you whether we can say that a mouse is depressed or not depressed, but we can ask whether it changes its behavior based on drugs that we know should actually alleviate depression symptoms. And the way that we evaluate them vary. They're generally responses to mild stressors in the environment. And in this case, I'm showing you what happens when we give the animal we put the animal in an environment that it can escape from, a small beaker of water, and if you give that animal Prozac or other drugs that are used as antidepressants, they will actually try to escape for longer. And if you evaluate mecamylamine, you can see that it works the same way. So here is the amount of time that the animal stays immobile. And what you can see is that if you give a nicotine blocker, the animal is immobile for much less time. And what you can see is that if we knock out these high-affinity nicotine receptors, remember those that are responsible for smoking, that effect is completely gone. Mecamylamine no longer changes the likelihood that the animal will be immobile. But what you can also see is that even at baseline, with no treatment with mecamylamine, these animals without the nicotine receptors are more likely to keep trying to find a way out of that inescapable environment than animals that have normal nicotine receptors. They look like their nicotine receptors have already been blocked. So that gives us a hint that perhaps blocking these nicotine receptors might be antidepressant in humans. So as a result of these studies and others, we have colleagues, Tony George and colleagues, who tried this in a human subject population. And what Tony George did was that he recruited patients who were already on Prozac or some other serotonin-regulating antidepressant, and they had not responded. And this is actually quite sad, and that is that about somewhere between 30 and 50% of individuals with depression don't respond to our current medications. So he recruited a subset of patients. He kept them on Prozac or whatever other antidepressant they were on, and he added mecamylamine. And he was able to do that because it had been used for blood pressure treatment in the past. And what he saw was that in this small group, he could actually improve their depression symptoms. And if he gave a placebo to these individuals who had been on Prozac, it had no effect. So at least in some small studies, mecamylamine can also be used as an antidepressant in humans. And we wouldn't have tried this if we didn't know that there was a rationale from these basic science studies and if we didn't have some data that it might work. In a very, very large trial recently, mecamylamine has not been shown to be the greatest add-on drug, so it may not be that this molecule is the one that is pursued for depression, but it does give an idea for future drug development. So the question is, same one I've been asking, where in the brain is this happening, and which receptors? So we went back to CFOS. Again, we looked at a marker, but this time of a decrease in neuronal activity because we're looking at the ability of nicotine blockers to cause this behavior. And we used both cytosine, which is also antidepressant-like in these models, and mecamylamine, and we saw that the place where we saw the best convergence in this case was the basolateral amygdala. And the basolateral amygdala is important because it's absolutely critical for stress responses. It's an area that's been shown to be hyperactive in subsets of depressed humans. And so we asked if we block nicotine receptors only in the amygdala by giving a local infusion, or if we knock down receptors only in the amygdala, can we recapitulate these antidepressant-like effects? And the answer is yes. We can block nicotine receptors with mecamylamine only in the amygdala, and we get decreased immobility. And if we knock down the beta-2 subunit locally, it has effects across models of antidepressant efficacy. So that leads to the idea that there's something about the signaling of the normal neurotransmitter, acetylcholine, in this brain area that's really important for regulating mood. So what we can ask is, can depression actually result from too much acetylcholine? And what we did was, again, to collaborate with our colleagues who are clinical investigators and to ask if we could see if there were any changes in acetylcholine signaling in the brains of patients with depression. So if depression is associated with an increase, there's a ghost trying to come into that door. It's been coming in and out. If depression is associated with increased acetylcholine, what would we see in an imaging study? So we can use a nicotine receptor binding tracer, and we can say, if there's a little bit of acetylcholine signaling in the brain, and we give this tracer to humans, that there should still be sites for it to bind in other receptors, and we should be able to image them. However, if acetylcholine signaling is elevated in the brain, it should occupy more of those receptor sites, and now when we put in the tracer, there should be fewer sites for it to bind to. And so that's exactly what we saw. In depressed patients who were actively depressed at the time that they were scanned, we saw less binding of this tracer throughout the brain than we did in the brains of people who had never been depressed. We have to do this in non-smokers because nicotine occupies exactly the same sites as acetylcholine, so we can't actually, we can't image people who are actively smoking. But this was suggestive that perhaps throughout the brain there was more acetylcholine occupying those receptors. Now, those of you who do these kinds of experiments could also say, well, hey, this could be a competition with acetylcholine, but it could also just be that they have fewer nicotine receptors, and that would be counter to your argument. That's exactly what we thought. So what we then did was we scoured the country for people who had brain banks of individuals who had died who were actively depressed, mostly from suicide, and individuals who had died at the same age but from accidents or other events that did not have to do with depression. And we asked, did they have any difference in the number of nicotinic receptors? We used exactly the same tracer that was used in the human imaging studies, and in those postmortem brain studies, there was absolutely no change in the number of nicotine receptors when we washed out all of the acetylcholine. And you can see we used also another psychiatric illness, schizophrenia, as a third control. And what you can see here is that it suggests that in the imaging studies that the decrease in binding to the tracer was likely as a result of the competition with the tracer. And I'm going to close, even though I have a few more slides to show you, with just a hint to all of you that really most of our ideas have in some way or another come up before. And so in the 1970s, a psychiatrist, Janowski, was able to increase acetylcholine levels in the brain by challenging with a blocker of the breakdown enzyme for acetylcholine, physostigmine. And he showed that when he challenged people with physostigmine, if they had a history of depression or even if they did not, he could increase depression-like symptoms in humans. And he suggested that that meant that if you have too much acetylcholine, you're more likely to be depressed, and based on the medications that were used at that time for depression, that having too much norepinephrine might make you manic, and so that this balance between norepinephrine and acetylcholine would be essential for depression. Unfortunately for Dr. Janowski, he proposed this right about the time of the Prozac revolution. And Prozac, as those of you who are pharmacologists know, actually works on the serotonin system. And because it became the dominant paradigm, serotonin was really pursued as the neurotransmitter in charge of depression, and Dr. Janowski's theory sort of got shunted aside. And I would like to argue that, in fact, we are not a bag of single neurotransmitter, that the balance of neurotransmitters across many systems are likely to be important for the kinds of complex behaviors that are really important for psychiatric illness. And so, in fact, it's quite likely, especially based on these human imaging studies, that we should be looking again at other systems, particularly the acetylcholine system, and asking, how does dysregulation of these other systems actually contribute to depression? And I'll close with one more slide of data to show you that actually we can block acetylcholine breakdown and particularly target acetylcholine breakdown to a particular brain area, the hippocampus, and we can see exactly the same thing that Dr. Janowski saw when he challenged individuals with physostigmine, but we can now identify the particular brain areas and the particular receptor subtypes that target that response. And in the interest of time, I'm now going to skip to my conclusions, which are that now we've got another area where we know the symptoms of a particular psychiatric illness can decrease the likelihood that folks can quit smoking. We have the same receptor that we've actually talked about with respect to nicotine reward and reinforcement, but in this case, instead of activating that receptor, in order to get antidepressant responses now, we want to block that receptor, and particularly we want to block it in the amygdala, as I showed you earlier. And if we do that, then we get potentially another insight into why some people smoke, even though we know that they're going to have a bad outcome. So I started by introducing the problem, which was the very large molecular group of targets for nicotine in the brain, and the broad expression of those nicotine receptors, as well as the large number of reasons and behavioral consequences of smoking. And then I have good news. The good news is that a combination of molecular genetics and of pharmacology, both in animal models and in humans, has actually, as a field, made very good progress in dissecting the nicotinic receptor subtypes and the brain areas that are responsible for these specific nicotine-dependent behaviors that contribute to smoking. So targeting these receptors is already a success story for rational drug design, particularly for smoking cessation, and it has resulted in an effective treatment for smoking. But we have a big challenge. We know that existing pharmacological agents used in humans, for the most part, don't target specific nicotinic receptor subtypes, and in fact, agents that are somewhat selective, like cytosine and mecamylamine, have different selectivity for human receptor subtypes. So we really have to do a better job of coming up with new safe molecules to target these different subtypes. And our hope is that medications that are targeted to these highly specific nicotinic receptor subtypes could be useful not only in smokers to help them quit or to motivate themselves to quit, even despite the fact that they're afraid that they might gain weight or they're afraid that they won't be able to control their depression symptoms. But maybe they'll actually be useful for individuals who don't smoke at all to treat depression or to help those who struggle with obesity to control their appetite. And I'd just like to close by thanking our funders, particularly the National Institute on Drug Abuse and the National Institute on Mental Health, NARSAD, and a Tobacco Center funding, as well as Tobacco Center as in Tobacco Treatment Center, not from the tobacco industry, no tobacco industry here. As well as our many collaborators in other laboratories and our current and former lab members who really contributed not only to the experiments that I spoke about here, but all of the planning of those experiments and the concepts and the design of those experiments. Thank you very much for your attention.